Record. Okay, recording, great. The lab, okay, that is right. I want to go, come on. Oh, no, I want you to do this. Okay, oh, come on. The lab, bio 1, lab 1, biochemistry 3 and 4. Okay, so do you guys see my, no, you don't see my screen. Come on. Okay, let's do it this way. Okay, you guys can see my screen now, right? Biochemistry part 1. So I ask you to draw all of the carbons, well, for the first time, I'm letting you do this. It's your responsibility to learn these things, but anyhow, it's up to you. I think if on a piece of paper, you draw these molecules, it's a better practice for you than you want to copy and paste from. Okay, let's go to chapter here, this is chapter 3, everyone. Is that right? So control F, and I want glycerol. Find it for me. Well, I'll go to the next one. Next one. Yeah, I want to see a picture. Come on. Oh, come on. You're kidding me. It does not have glycerol? Or maybe it does, but these are proteins. There are fats. These are amino acids, DNA, nitrogenous bases, glucose, DNA again. Well, here we go. Okay, here is a proteins and lipids macromolecules. Here is assembly of this assembly. Maybe I should do this at the end of the lecture, because disaccharides, polysaccharides, cellulose, nucleic acids, DNA, ATP, proteins, peptide bond, motifs, yes, domains, chaperones, denaturation, lipids. Okay, these are steroids, fats, triglycerides. Here we go. So a glycerol molecule, he did not do it. Okay. Okay, the only thing is missing is it's a glycerol molecule. Everything else is in there. So, but the fatty acid, this is a fatty acid. You guys see that? So the way you can do it, and I will probably take it. You can copy this, control copy, or right click. Do you guys follow what I'm talking about? No. Okay, right click, copy. Do you guys follow what I'm talking about? And then I go to this one. I prefer you would draw it by hand, but for the first time, I'm taking and then control paste. Right? So then you click on it, and then you want to crop. You go to format, and then you crop. Some of you are familiar with. So a glycerol molecule is this one right here. So you want to bring it up. There is an attached to the oxygen, there is one hydrogen as well. So your textbook failed. I should copy it from another document, and I will put it in your lecture notes, and I will put it up. So I got to write a note, because there could be with me changing textbook, it could be other pictures that are missing. So I got to get a glycerol, and I will put it in this PowerPoint presentation. So you do have a molecule of glycerol just by itself. Same thing, and now draw, this one, this is supposed to go down here, but anyhow, and then draw a molecule of fatty acids. You guys follow what I'm doing? Another paste. So fatty acid, 8 carbons. Do not worry about 8 carbons. Again, you go here, you go to format, you go to crop. And of course, after today's lecture, you had Bio 1, you had Chemistry 3A. So you crop up to here. At the other end of fatty acid is not like that. It is pretty much like this. Okay? I will put a picture of a fatty acid for you guys in here, unless, and then whatever else I see that is missing. And the next one, you don't crop anything, you just paste the picture. Do I make sense? Then you have to print this and bring it to the lab. If it is black and white, is perfect. You have color print, you want to print color, it is perfect. Anything you do is fine with me. Any question? And the rest of the lab is like that. I'm going to go over the answers. Monday, we don't have class. And Wednesday is the last. Okay, I try to finish everything by today. We'll try. Does anybody have any question about this? Let's go over chapter.
Actually, can I ask, Professor?
Yes.
Is it okay if we like just write out the question and then we draw out the
Yes, I like that better.
Yeah.
I said
Okay.
you just joined us or what? I
Oh,
said
no,
that
yeah,
I like
I've been
that
in the
better.
beginning.
For the
Okay.
first time, I'm allowing, for the first time since I've been teaching this class, I am allowing for you to copy paste. Of course, you have to print it and bring it to the lab, but you want to do it by hand? Perfect. I want all of the hydrogens and carbons drawn.
Okay, thank you.
Yes. Okay, so we talked about these things last time. The carbon is the framework. Does anybody know the enantiomers? Did I talked about this? Molecules are mirror image of each other. Did I talk about this or no? Yeah, I think I did.
Yeah,
So
you did.
yes, thank you. And then I said, make sure you know this table. A lot of multiple choice questions in the exam, and also and next week quiz. After next week, you have an exam. After next week quiz, you have an exam. You already had a quiz today. I tried to grade it and put it in Canvas ASAP. Okay, the first group of macromolecules or building block that your textbook is talking about: carbohydrates, nucleic acids, proteins, and lipids. Yes. Assembly of disassembly molecules can break down. Okay, I didn't go over this. This is important. Let me. Okay, great. Let's go. So molecules, chat question, is it okay if we write these instead of, yes, she already asked. Okay, dehydration synthesis, it means when you have a molecule and you are taking a water molecule out of it. You have two molecules, sorry. You have two molecules, one molecule here, another molecule here. You combine the OH of this molecule and you will see it a lot. And the hydrogen of that molecule form water molecules. So the process is called dehydration. Then if you have a molecule, you have one molecule like this, and you add water to it to break that bond, the name of the process is called hydrolysis. Lysis at the end of the word means what? Those of you who study your prefix, your root words, I didn't grade your quizzes. I don't know how well you study them. Lysis at the end of the word means break down. So you're breaking this molecule to two molecules. Opposite of it is dehydration. So carbohydrates is the first building block we are going to talk about. The molecules with a ratio of 1 to 2 to 1 ratio of carbon, hydrogen, oxygen. It means for every one carbon, there are two hydrogens. For every one oxygen, there are two hydrogens. So empirical formula is CH2ON N number. So, and then this is a covalent bond, right? You guys know that? That covalent bond can be double two, not with hydrogen, but with oxygen, right? You cannot have a carbon and a hydrogen with double covalent bond. That cannot happen. So covalent bonds hold much of the energy. Carbon are a good source of energy for storage molecules, and examples, sugar, starch, and glucose. I'm hoping you write down in your notes that 1 gram 1 gram of carbohydrates gives you 4 calories. Let me write it down so you guys know, and I have it in my notes. So 1 gram of CHO carbohydrates gives you 4 calories. Oh, come on. I hope I spelled it correctly. Yes, I did. Okay, that's a number I would like you to know. Control S. So it will be in your next quiz exam. So that's it. And then we'll talk about the other one. Mono, it means one. Saccharide, it means sugar. So simplest carbohydrates, you dealt with it in the lab today. Glucose is a single sugar molecule with 6 carbon, play important roles. Glucose C6, the empirical formula for glucose is C6. It means it has 6 carbon, 12 hydrogen, 6 oxygen. You remember 1 to 2 to 1 ratio? You remember that? I just talked about it. Okay. The fructose is a structural isomer. It looked like glucose, but it is different than glucose. I hope the pictures are there. Galactose is also a stereoisomer of glucose. It's an isomer of glucose, C6H12O6, but the place the oxygen and hydrogens are on carbon number 1 and number 4, or number 1 and 4 is different. So here it is a glyceraldehyde. Holy cow, we talked about, yeah, this is it, glyceraldehyde. So if you want to put it's a little bit different. I'll put glycerol in there for you guys. Here's a ribose. Ribose has 5 carbons. You see that in DNA and RNA. You guys with me? Deoxyribose, it has this, look, the difference between, one of the differences between DNA and RNA. DNA has deoxyribose. That's what they call it, deoxyribose nucleic acid. Look at carbon number 2. It has just hydrogen. But look at ribose on the RNA. On carbon number 2, it has the oxygen attaches to another hydrogen. Glucose, fructose, and galactose. All 3 of them they're all monosaccharides. All of these are monosaccharides, all of them. This one has 6 carbon. This one has 6 carbon. This one has 6 carbon. This glucose has 12 hydrogen. Fructose have 12 hydrogen. Galactose have 12 hydrogen. 6 oxygen, 6 oxygen, 6 oxygen. So they are isomers of each other. Okay, carbohydrates usually in a chain. If I have this, I better not spill my tea, then oh boy, I'll be in trouble. In the morning, I drank coffee for you guys. Now I'm drinking tea for you guys. So this is like this, and then you make it a ring. That's what Mother Nature does. Mother Nature can do this with the help of enzymes. This is it. This is a straight chain of carbohydrates. Now it is in a ring format. This is what I would like you to draw in the in your lab biochemistry part 1. He's saying, look, when he's saying that this one, well, I don't want to draw this one, I want you to draw this one for me. So on one corner there is oxygen attaches to that carbon. If you look at all of these carbons and hydrogen, you see carbon here has 4 covalent bonds, 1, 2, 3, 4. This oxygen has 2 covalent bonds, one to the carbon and one to the hydrogen. They didn't draw it in a dashed line, but that's OH means a covalent bond between oxygen and hydrogen, between oxygen and hydrogen. Okay, so there is a difference. I would like you to know, you should know, not I would like you know, you should know there is a difference between alpha glucose and beta glucose. This is not galactose, this is not anything else. This is alpha glucose and beta glucose. What is the difference? Carbon number one, everything else is alike, but they highlighted for you carbon number one, the OH is on top for beta. And for alpha, the OH is on bottom. The beta is found in cellulose, like you eat celery. There is a certain portion of celery is indigestible. Cellulose, beta. Mashed potato, alpha. Okay, everybody, I hope I'm making some sense. So I am giving you the answer to the quiz question, to the essay question. Here they are, structural isomers, what happens between glucose, fructose, galactose. I'm not too crazy about this, but please make sure you know it. And then the disaccharides, it means two sugar molecules combined together. They went through the process of dehydration. One sugar molecule, they're combined together through the process of dehydration. They became two sugar molecules combined together. So right here, the alpha glucose with fructose, which is sucrose, right? This is a disaccharide. Sucrose is a disaccharide. Look, they're bound together right here. The oxygen between them, between a sucrose, between a fructose and a glucose. Maltose, O-S-E at the end of the word, glucose and glucose. And then we have lactose, the one in milk. Is it going to come up? No, it does not want to come up. I will put it up. I better draw it down. Okay, I will put up lactose in your PowerPoint. So lactose is glucose and galactose. Where is Amir galactose? Right here, right here. Look, oh, look how different is glucose and galactose. Of course, fructose is a pentagon. Pentagon, it means it has 5 sides. You should know these terms. Pentagon means it has 5 sides. Hexagon, it has 6-sided. It's a molecule with 6 sides. Hexa, penta, 5 sides, but it still has 6 carbon. Now look at galactose and glucose. Galactose on carbon number 4, they number them. Carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6. On carbon number 4, the galactose, the OH is on top, but on glucose, the OH is on the bottom. It's not alpha and beta. Remember that? Alpha and beta is carbon number 1. Okay, I hope I'm making some sense. Maybe you should watch these videos more than once. There are other, the polysaccharides, many of them attached together. Many units, many monomers. This is a monomer. This is a monomer. When there are 2 of them together, they call them disaccharides. But there are many of them attached to each other. It's called polysaccharide. So long chain of monosaccharides links through dehydration synthesis. We already talked about that. Energy storage. Plants use starch. Animals use glycogen. Yes, all of these are great quiz questions. And I'm hoping you would know. Structural supports. In plants, you have cellulose, right? And then arthropods and fungi, they have chitin. I hope those pictures are here. Maybe chitin is not here, but I'll have to bring it and put it up for you guys. So here is a glucose molecule, alpha. They call it alpha linkage. This is the one that is found in rice, mashed potato. and sometimes they can branch. Still, it's alpha linkage, but they can branch. So the difference is the polysaccharides inside of human is always branched. That's what they call it, glycogen. However, the carbohydrates attached to each other in plants, like mashed potato, rice you eat, they are either straight or they're branched. They're either this or this, branched or straight. However, in animals, glycogen, we call it glycogen, they don't call it starch. Sometimes you see it in your quiz or exam that they call it animals' starch is glycogen, not cellulose. Animals' starch is glycogen. Okay, not DNA, not nothing else. All of the other, not protein, no. Okay, I better save that for the time when the throat gets dry. Okay, are you ready to move? And they are showing you the branch one, and of course the straight and branch one. Amylose is the one that is straight. Okay, cellulose is a polysaccharide, but it's a beta linkage. It's not alpha, it's beta. Cellulose. Celery you eat. The wood, the plants, the chair here, the paper. These are all cellulose. It means they are beta linkage. The most abundant molecule on planet Earth is cellulose. More than any other molecule on planet Earth. And you should eat it. You should have it in your diet. Fruits and vegetables, they have cellulose. You cannot digest it, but it's good for your, it allows bacteria to grow in your gut. Good bacteria, of course. When the good bacteria grows in your gut, then you can have a good defecation. I hope I'm making some sense. Okay, any question? The next one is nucleic acid, the blueprint. I talked about this last time and it was a quiz question. I hope you got it. The blueprint for all living bacteria, tree. The mushroom, it's in your fridge, all living amoeba, all living organism, the blueprint is right there, DNA. Okay, based on the DNA, then I have black eyes, black hair, dark complexion, whatever it is. Height, my height is based on DNA. DNA decides. Okay, so polymers are nucleic acid, monomers are nucleotide. I hope there are pictures of them in here. If they are not, I gotta get you guys those pictures. Right now, I gotta get glycerol, lactose, and chitins pictures in here because we do need them. Sugar plus phosphate, so and nitrogenous base, which make up the monomer. Okay, sugar is deoxyribose in DNA and ribose. We already discussed that. Nitrogenous bases include purines. Purines are adenine and guanine. Okay, those two, pure gold, easy to remember, pure as gold. Purines are adenine and guanine. And pyrimidine, which is a longer term, it means thymine, cytosine, and uracil. Anyhow, I will put that in there. Phonemics for DNA and RNA. I will put that. Okay, you guys. And nucleotides connect by phosphate together right here. So here's a phosphate group. Here is a ribose. RNA is OH. This one, carbon number 2, 2' they call it. Yes, it is true, 2'. And then this is carbon number 1, carbon number 2, carbon number, not carbon, the corner, I don't why he's calling it number 3, I'm not sure. But anyhow, I do not number them like that, but the other carbons in other parts of the molecule is carbon 1, 2, 3, 4, 5, 6, but the carbon on the sugar molecule, the ribose or deoxyribose, is called 1', 2', 3', 4', 5'. The 5' attaches to the phosphate. And then the 1' attaches to 5' and 3' supposed to be attached, as you see it later on. Anyhow, this one is attached to the nitrogenous base. This is the nitrogenous base. It has 2 rings. Purines have 2 rings. Pyrimidine, you will see they have 1 ring. We talked about it. Purines have 2 rings. Pyrimidines, they have, which are there are 3 of them, they have 1 ring. Here is a nucleotide structure. Here is a DNA, phosphate, sugar, phosphate, sugar, phosphate, sugar, and the nitrogenous bases. Here attaches 5' carbon, number 5, and so on and so forth. Okay, great. Purines, you remember I said they have 2 rings? For the lab, practical, and also for the lecture exam, 2 Wednesdays from today, not next week, the week after that, I will put these pictures in the exam, and you should recognize them, not by this name. If you want to write down guanine, sure, write it down guanine. You want to write down adenine, sure, adenine. But if I put guanine in there and you write down adenine, I will not give you credit. I would like you to write down purines. Purines, because why? Because it has 2 rings. All of these are nitrogenous bases. Nitrogenous bases are broken down to 2 groups: purines and pyrimidines. Purines, they have 2 rings. Pyrimidines, they have 1 ring. Do you have to recognize them separately? No. I know somebody's going to ask me that. Uracil, of course, I'll cover all of these names. And then, oh, Amir, do you have to know uracil from thiamine? No, you don't. Just tell me it's a pyrimidine. Okay? And cytosine, do not mix it up with, we have an amino acid, cysteine. Do not mix it up with cytosine. So cytosine, thiamine, and uracil, and the good thing about your textbook, they said where they are found. Okay? So deoxyribose nucleic acid, DNA, encodes, I hope I don't go over time. Oof. Encodes information for amino acids, yes. Double helix, yes. And A with T and C with G, as you can see it in DNA. You probably studied it before. RNA, ribonucleic acid, and contains ribose instead of deoxyribose. Here they are. The difference between, another difference between DNA and RNA. DNA is double-stranded and RNA is single-stranded. Okay, so it would be nice for you to make a table for yourself to write down the differences between RNA and DNA. Adenosine triphosphate, I can do this because of that molecule, ATP. ATP is a source of energy inside of your body, all living organisms. ATP and GTP, you're going to learn. But right now, let's stick with ATP. Okay, so primary energy source of occurrence in the cell, nicotinamide, adenine dinucleotide. I'm not too worried, and FAD, do not worry about them now. When we get to cellular respiration, I'll talk about them. But for now, do not worry about it. And here's the structure of ATP. It has 3 phosphate. And the combination of the adenine and ribose, sugar, ribose, and adenine, they're covalently bond together. It's called adenosine. Adenosine It means combination of adenine and ribose. Adenosine tri, it means 3, 1, 2, 3, phosphate. And then when ATP breaks down, ATP, 3-phosphate, breaks down to 2-phosphate, it releases energy, you can move, you can blink your eye. Proteins, these are functions of proteins. As semester goes along, we will talk about all of these functions. Okay, so we will talk about these functions. Here, protein characteristics. This is a general characteristics of an amino acid. I like it. They draw it big and huge. It has an acid end. You remember the functional groups? I asked you to study them. This is an acid end. This is a hydrogen end. This is central carbon. This is amino end. And this is the R group. There are only 20 different, 20. Remember that number. There are only 20 different amino acids inside of our body. What makes those amino acids different? This right here. R group, radical group, they call it. Composed of one or more long unbranched chains. Chain is polypeptide. Amino acids are monomer. Yes. For proteins, amino acids are monomer. For DNA, RNA, monomers are nucleotides. You should remember the monomers and polymers. And protein is a polymer. Amino acids, central carbon, amino group, carboxyl group, which is an acid. That's what they call them, amino acid, singular hydrogen, yes, and variable R group, radical group. Here are the amino acids. You guys, I don't expect you to memorize the structure of these amino acids, but I expect you to recognize the names. You should recognize the names. Alanine, valine. Darn it. Okay, I don't like it when they're so small. I cannot read it. So anyhow, I made it big enough for you guys, I hope. Let's make it a little bit bigger. Okay, it's fat enough. Control S. Okay, let's go over the names so you can. Alanine, I can see it a little bit better. Valine, isoleucine, leucine, glycine. In a multiple choice format, I say which of the following is an amino acid? Which of the following is not an amino acid? Serine, threonine, and it is one of the amino acid. Anyhow, it's not there. Asparagine and glutamine. I don't know why. Glutamic acid. No, methionine has. Like a glutamic acid, arginine, aspartic acid, lysine, and then you have histidine, tyrosine, tryptophan, phenylalanine, proline, methionine, and cysteine. There are 2 amino acids, you guys. There are 2 amino acids that they have sulfur. One of them is methionine. Look, the sulfur. But it usually does not react with other molecules. But this one, cysteine. Cysteine reacts. The sulfur forms disulfide bond with another molecule. With another possibly cysteine. Okay, so there are a few of these amino acids. I would like to know something about them. Cysteine, you should know it has sulfur and can form disulfide bond. Methionine has sulfur, but it does not form disulfide bond. Proline, you know, it forms a ring between the central carbon and the R group. Okay. The simplest of all amino acid is glycine. Here we go. Has a just a. The R group is just hydrogen. Okay. So those are the 3 or 4 amino acids. I would like you to know something about them, and the rest, of course, you have to know the name. Okay. What is a peptide bond? Peptide bond, first of all, is a type of covalent bond. You took a quiz over today. It shares electron. So the hydrogen, these two hydrogens of this amino acid from the amino group combine with the oxygen of the acid group of this amino acid, and they're combined together, and that is the peptide bond right here. I'm pointing at it. This covalent bond between nitrogen amino group of this amino acid and the carbon of the acid group of this amino acid form a peptide bond. A peptide bond only happens in amino acids, nowhere else. So this is a dipeptide molecule. The name of the molecule is dipeptide. The name of this molecule is amino acid. The name of that molecule is amino acid. Okay, the shape of a protein determines its function. Primary sequence is just like this. Oh, please go aside. So primary is the amino acids are in a chain like this. Okay, secondary structures, there are two different types, and helix and like a pleated sheet. I hope the pictures are there. Helix like a spiral. Okay, so tertiary structures, they are globular proteins stabilized by a number of forces. Final level of structure of proteins. I know these things do not make any sense to you guys. I hope I have pictures. If not, I will get the pictures and put them in here for you. And quaternary is like found in hemoglobin. Tertiary is found in myoglobin. Myoglobin is found in your muscle cells. It stores oxygen. And hemoglobin is found in your red blood cells. It transports oxygen. It cannot hold on to them for a long time. Here we go. Primary, two types of secondary, spiral, helix, we call them. And then pleated, like those ladies or those Scottish people were that is pleated skirt. You all know what I'm talking about, pleated skirt. The secondary is pleated. Do you guys see this? And then you have the tertiary look like this. These molecules can come together and form a tertiary molecule. These two can form, which is your myoglobin. Okay, and then look, this molecule, which is found in your red blood cells, hemoglobin, it has one tertiary, two tertiary, three tertiary, four tertiary combined together. It's called quaternary. That's how things are. Okay, interactive proteins molecule. You might say, how does this fold? The fold are because of hydrogen bond, because of disulfide bond. You remember cysteine, we talked about it. They fold, they become helix, or they become pleated or quaternary because of ionic bond. The van der Waals one, we didn't talk about them. It is almost like a hydrogen bond. It's a force. Okay, but anyhow, and hydrophobic exclusion. So they can form, they do not hydrophobic, it means does not like water. This end of the molecule, this inside of the molecule does not like water. Okay, additional structure characteristics, motif. Yes, I would like you to know what is a motif and domains, a common elements of secondary structure seen in many polypeptide. They're talking about motifs. We are talking about the protein molecules. Useful in determining the function of unknown proteins. And then domains, functional units within a large structure. You have a huge molecule, but only a little bit of it right here is functional. That's what they call a domain. And then most proteins made up of multiple domains that perform different parts of the protein's functions. So anyhow, I hope you know the difference between motif and useful determining function of the unknown protein. Here is a motif. You have either the helix or you have the pleated sheet. But here you have a domain, huge molecule. Only this portion of the molecule is functional. Only that portion of the molecule is functional. And only this portion of the molecule is functional. The rest of this molecule is not functional. That's what the difference between domain and motif is. Chaperones, you've heard of the term chaperones. When you have a younger siblings or younger child at school, they ask for parents to be chaperone. It means it escorts the molecule. Proteins do that inside of our body. They escort the molecules. So originally thought they made up of proteins folded spontaneously. No, chaperones proteins help proteins to fold correctly. Yes, deficiencies in chaperones proteins implicate certain diseases, cystic fibrosis, and other diseases is called. Here is a protein molecule. Okay, the other protein molecule, it will go inside of chaperone and become folded properly the way that your body can use it and get out of chaperone, corrected folded protein, because your body cannot use this protein. It's just not folded correctly. I hope I'm making some sense. But it goes inside of the chaperone and chaperone folded correctly. Now your body can use it. Many growth hormone, insulin, many proteins or molecules are. Denaturation, it means when you have an egg, you crack the egg and fry it, you denature the protein molecules. The proteins molecules did not vanish. They did not disappear, but you denature them. If they were like this, the protein molecules in the egg, now they're like that. They call it, you denature them. So proteins loses its structure, the fold, and also its function, of course, yeah. After you fry an egg, you cannot make that egg cannot become a chicken anymore, a chick. Am I making sense? I hope. Due to environmental conditions, these are the factors that can cause denaturation. What are they? pH. On a protein molecule, you pour a strong acid, it will denature it. You put a strong base, it will denature it. Temperature, as I said, fry egg, and then ionic concentration of solution. You put the ionic, a very salt structure solution on it, and then it folds. Okay, protein denaturation. Sorry about that. Sorry about that. And then here are, please, scientific thinking. Read them, know them. Lipids, the last one. Lipids are not, they're huge molecules, but they are not polymers. Remember, polymers, we said they are made up of, like, do you have rosary? You know what I'm talking about? The rosary. I wish I would bring one. Rosary is a polymer. You guys know what a rosary is, right? Everybody? Rosary is a polymer because it's made up of those little beads. Lipids are not polymers, even though they're huge molecules. They're not polymers because they do not have repeated units. For carbohydrates, repeated units was glucose, for example. For protein, it was amino acid. For DNA, RNA, it was the nucleotides. Okay, so loosely defined group of molecules with one main chemical characteristics. They are insoluble in water. Okay, high proportion of nonpolar carbon-hydrogen bonds causes the molecules to be hydrophobic. So fats, oils, waxes, terpenes, steroids, and even some vitamins can be. Here we go, steroid. Okay. And then fats, triglycerides. I will talk about composed of a glycerol molecule and 3 fatty acids. I will put it up in here for you guys. Fatty acids needs to be identical. And then chain length varies. Saturated, it means all of the hydrogens. I'll show you in a picture. Saturated fatty acid means all of the carbons have hydrogen attached to them. Unsaturated fatty acid means a few of the hydrogens are missing. That's what it is. Trans fats produce industrially. Okay, right here is a fatty acid. This is a saturated fatty acid attached to a glycerol molecule. I will put up these for you. This is unsaturated fatty acid. Still, look, carbon has still 4 covalent bond. This carbon has 1, 2, 3, 4 covalent bond around it. But this is unsaturated fatty acid. This is unsaturated fatty acid. This is unsaturated fatty acid. Saturated fatty acid, saturated fatty acid, saturated fatty acid. Okay, and of course, the one unsaturated fatty acid look like this. The one saturated fatty acid, you're straight chain. So phospholipid composed of glycerol, 2 fatty acids, nonpolar tails, a phosphate group, polar head, forms all biological cell membrane right here. This is a phospholipid. So you have a choline group right here. This is called the green. It's called choline. Very good. It's listed. A phosphate group and a glycerol group, 3 carbon. And then 2 fatty acids. All of these fatty acids are saturated, but in real life, you know, the one of the fatty acid is kinked. It's they have some unsaturated fatty acid in here, so the molecule is kinked. Straight molecule, kinked molecule. We call it kinked. So the head of fatty acid, we call this head, is made up of choline, phosphate, glycerol. The tail is made up of fatty acids. Any question? And the head is love water. They like water. The tail do not like water. Hydrophobic, hydrophilic. Okay. So here is a membrane formed, and we will catch up on that. Cell membrane look like this. We will catch up on that. And I am done. They should give me a medal for finish all of this in 1 hour. But listen, I'm going to put this PowerPoint is going to be a little bit different. I'm going to add more slides to it. And you guys should download the new one of chapter 2. It might happen throughout the semester because I go through and then I see things. Any question? Listen, everyone, please, you go to gradebook, you see 0 or you don't see any score for your metric system. Please write it, print it, whatever you want to do, bring it to the lab, because next Wednesday is the last date that I accept metric system. Next week, bring biochemistry 1 and biochemistry part 2, both of them separately. And those students who staple them together, both biochemistry 1 and 2 and metric system, I know they did not watch these videos. Okay, I'm running out of battery on my laptop. Any question, you guys? Okay, I guess not. I guess I was